All right, so in this video note, we're going to quickly review how to use the genetic code, which is what you've seen already before, something like this, and determine the amino acid sequence from a gene. So these are the exact same thing. They both are the genetic code. They are just written in a different format. You can use it both, and they both say the exact same thing. So let me show you. So if I have a strand and I'm looking for UAG, that's my mRNA UAG. So UAG means stop. Okay, I'll show you how to use this over here, but UAG, stop. They both mean stop. So you can use them the same thing. So it's just a preference. So first I want to talk about how we can use this version of the genetic code. So here's our DNA sequence. AAC, GCT, AGG. And so you remember, we have to read the codon. So that means we have to read every 3 letters. So we first have to figure out what our mRNA strand is. So A, what would be the nitrogen base? Can you pause and think? Why don't you pause and see if you can figure out the whole mRNA strand without me. All right, so it should be this: UUG, CGA, UCC. Okay, now that we have our mRNA, now we can find out what amino acids go with those codons. Remember, one codon has one amino acid. Now, you can have one amino acid has several different codons, but you need to use the genetic code to figure out which one is which. So we have UUG. So U means we're going to be in this box right here, okay? And so and then UU, which means now we're going to just be in this box. So this is the first letter. This is the second letter. The third letter is over here. And so it'd be G. So now we're in this box all the way over here. So that should be our first amino acid, leucine. Okay. The next amino acid is CGA. See if you can use the genetic code and figure it out on your own. CGA. All right, so we should be looking at this box only, all the way across, and then G. So now we're only looking at this box. So now we're only looking at right here, and all of them say the same thing, and that would be arginine. All right, the next one is UCC. So we're back up into this box again, U, and then C, and then C. So that would be serine. All right, so that's how you use this genetic code setup. Now I'll practice using this one just for refresher from what we had in the video notes a couple days ago. So again, here's our DNA sequence: AAG, CTG, ACG, ATT, CGG. So that's our gene sequence. So that's our DNA that codes for a specific protein. So now we need to remember the mRNA. We first have to transcribe it into our messenger RNA before we can start translating it. So see if you can pause and do this on your own, just verbally. All right, so A goes with U, uracil. Remember, RNA has uracil, not thymine. And then it should be GAC, UGC, UAA, and then GCC. All right, so now we need to use the genetic code to find out what amino acids go with each codon. So UC. So when we say U, that means we're only working with this bracket. U means now we're only working with these. And then C is right here. So that means our amino acid is phenylalanine. All right, then GAC. So now G will be working with just this bracket. A, so now just this. And then C. So now it would be this amino acid, which is aspartic acid. All right, so UGC and UAA and GCC. Pause and see if you can do it with this type of genetic code setup. All right, so UAA, UAA. So UAA would be stop. It would be the stop codon. So then you don't do GCC because you stopped. It stopped. The amino acid sequence is done because we reached our stop codon. All right, so now what I want you to do is open up the transcription translation Google Doc, which has 3 more practice problems that are a little bit longer. You choose which genetic code setup you want to use, either the circle or the box, and complete the Google Doc practice, and then there's some other review protein synthesis questions as well.